Bismillahirrahmanirrahim. Assalamu alaikum dear viewers. I am Dr. Muhammad Usman, Professor of Physiology, and in today's video we will discuss the smooth muscle physiology. Now you know that smooth muscle is one of the two involuntary muscles in our body, the other being the cardiac muscle, and it is non-striated. Smooth muscle is widely distributed in our body. It is present in the walls of the hollow organs like urinary bladder and stomach, and it is also present in intestine and in blood vessels, in eye, in glands, in uterus, in skin. So widely distributed. Now its functions are according to its location. It causes regulation of the blood flow, it causes mixing of the food, it causes dilatation and constriction of the pupil, it propels urine, and at some locations it is autorhythmic, which means that it does not need any external stimulation to contract. Now if we look at the structure of the smooth muscle, then the smooth muscle cells are fusiform in shape or oval in shape. They are clearly non-striated, but having said this, actin and myosin, they are present in smooth muscle cell, as you can see here, but they are not arranged in sarcomeres. And the fibers are smaller than those in the skeletal muscle, about 30 times smaller. If we talk about the actin filaments, then tropomyosin is present, but there is no troponin, and also the amount of actin is much more than myosin. Then in the smooth muscle, caveolae are present. These are the indentations in the sarcolemma, and they may act like T tubules. There is one nucleus. Dense bodies are present, as you can see here. These dense bodies are present, and they are analogous to the Z lines of the skeletal muscle. They give attachment to the actin filaments, and they also give attachment to intermediate filaments, as you can see here. Intermediate filaments like vimentin, desmin, etc., which attach to the other intermediate filaments and dense bodies and serve to transmit the contractile force to sarcolemma. An important characteristic of the smooth muscle is that its myosin head has got a slow ATPase activity if we compare it to the skeletal muscle myosin head. Also, there is little sarcoplasmic reticulum in the smooth muscle. There are two major types of the smooth muscle, that is visceral or single unit or unitary smooth muscle, and as the name indicates, such smooth muscle is present in the viscera, and it is called single unit because the whole sheet of the muscles, as you can see here, it contracts as a single unit. Only a few muscle fibers are innervated in each group, and the impulse spreads through these gap junctions, as you can see here. It is often autorhythmic. The other type is multi-unit smooth muscle, and here you can see the locations of single unit smooth muscle or visceral smooth muscle. It is present in the walls of hollow organs like this urinary bladder, stomach, intestine, etc. So it is widely distributed. So this is the multi-unit smooth muscle, and in this the cell or groups of cells, they act as independent units, and usually each unit receives its own nerve supply. This is the autonomic nerve supply. Then about the distribution of multi-unit smooth muscle, it is less distributed. For example, there is erector pili muscle, which is present at the base of the hairs which are present on our skin, and when this muscle contracts, this tiny smooth muscle contracts, the skin hair, they are erected like this. Okay, you can see here. Then it is also present, multi-unit smooth muscle is also present in our eyes, in the pupil, and it causes constriction or dilatation of the pupil. Now let us discuss the mechanism of contraction and relaxation of the smooth muscle, which is very important. First of all, we will discuss its contraction. Now the first step in contraction is increase in the cytosolic calcium. This increase in calcium in smooth muscle cell, it can occur in response to the nervous, hormonal, chemical or physical factors. So there are a lot of stimuli for the contraction of smooth muscle. A simple stretch can lead to increase in the calcium in the smooth muscle. Then cold, autonomic nervous system stimulation. And it is noteworthy that if sympathetic stimulation, that is norepinephrine, it causes contraction at a certain place, then parasympathetic stimulation, that is acetylcholine, will cause relaxation at the same place, and vice versa. The major source of calcium for the smooth muscle is extracellular fluid, but action potential may be transmitted through the caveolae to the sarcoplasmic reticulum and cause release of calcium from the sarcoplasmic reticulum into the sarcoplasm, but its amount of this calcium will be small. So, in any case, the first step is increase in the calcium, as you can see here in this figure. Now, once the calcium is inside the cell, it binds to a protein which is called calmodulin, okay, calmodulin, CAM, calmodulin, which causes activation of calmodulin dependent myosin light chain kinase, MLCK, myosin light chain kinase enzyme, and this kinase enzyme, it causes phosphorylation of the myosin head, okay, as you can see here, it causes phosphorylation of the myosin head, and as a result of this phosphorylation, there is increased ATPase activity of the myosin head. Now, once the ATPase activity of the myosin head is increased, it can cleave ATP to ADP and phosphate, and now the myosin head can bind with the active sites of the actin filament, and it can cause a power stroke, pulling the actin filament in one direction on one side and in opposite direction on opposite side. In this way, the smooth muscle cell can contract up to 80% of their lengths, in contrast to the skeletal muscle cells which can contract only 30% of their lengths. Now, as long as the myosin head is attached to the actin filament, contraction will be maintained. Now, we will discuss the mechanism of relaxation in the skeletal muscle, and like contraction, the relaxation can occur in response to a large number of stimuli. For example, autonomic nervous system stimulation, and again if sympathetic causes contraction at a certain place, then parasympathetic will be causing relaxation at the same place. Then its relaxation can also occur in response to the local metabolic factors such as hypoxia, decreased oxygen, hypercapnia, increased carbon dioxide, acidosis means increased H ions, and increased potassium acid, potassium, lactic acid, adenosine, and then nitric oxide. They can also lead to the relaxation of smooth muscle. Now, all of these factors, the first step they cause in the relaxation of smooth muscle is decrease in the cytosolic calcium. Okay. So, the pump which moves calcium from the sarcoplasm into the sarcoplasmic reticulum, it gets activated, and also the other pump which moves calcium from the cytosol into the extracellular fluid, it is also activated. So, the calcium is pumped outside or into the sarcoplasm, and as a result of which the cytosolic or sarcoplasmic calcium is reduced. So, once the calcium is reduced, it gets separated from the calmodulin, and now there is decreased activity of myosin light chain kinase enzyme. Okay. Myosin light chain kinase, which was calmodulin dependent, its activity is decreased, and there is increased activity of an other enzyme which is called MLCP, okay, myosin light chain phosphatase enzyme. As you can see here, when the activity of myosin light chain phosphatase is increased, it causes dephosphorylation of the myosin head. Okay, it causes dephosphorylation of the myosin head. So, there is decreased activity, there is decreased ATPase activity of the myosin head. Once the myosin head is dephosphorylated, its ATPase activity is decreased, and it means that it cannot now cleave ATP to ADP and phosphate. So, now there are two possible scenarios: either the cycling stops and relaxation of smooth muscle occurs, or there is sustained contraction. Okay, there may be sustained contraction if the myosin head they remain attached to the actin, and it occurs due to latch phenomenon. So now let's discuss what is latch phenomenon. It is a mechanism which allows the smooth muscle to maintain tonic and forceful contraction. Okay, it is a mechanism which allows the smooth muscle to maintain tonic and forceful contraction for prolonged periods of time with added benefits. Actually, the smooth muscle myosin head, it has got low or decreased ATPase activity, as we have discussed before in this video, as compared to the skeletal muscle myosin head. Further, the smooth muscle myosin head, it also needs enzyme MLCK, myosin light chain kinase, to increase its activity, and MLCP, myosin light chain phosphatase, to decrease its activity. Due to this, there is slow cycling of the myosin head in the smooth muscle cell. Now, this produces three effects. Number one, there is increased force of contraction of the smooth muscle, because due to slow cycling, the myosin heads they remain attached to the actin for prolonged periods of time, and since the force of contraction depends at any time on the number of myosin heads attached to the actin filament, this will lead to the increased force of contraction. It is about 4 to 6 kg per centimeter square as compared to 3 to 4 kg per centimeter square in the skeletal muscle. Then of course, less ATP is used in the smooth muscle, because we know that ATP is needed only when we have to cause detachment of the myosin head from the actin, and this event occurs less in the smooth muscle, and also less excitation is needed as if we compare it with the skeletal muscle. So less energy will be used. Hope you would have understood about the basics of contraction and relaxation of the smooth muscle. Please consider subscribing to this channel. Happy learning, stay blessed, and Allah Hafiz.